All right, so we can continue with chemistry. I know we're one lecture behind. That's probably due to the covering the syllabus in depth. All right, so when we last left off, we were talking about reactions and bonds. And remember, covalent bonds. So covalent bonds are when two atoms share electrons. So here we have two hydrogen atoms. They're forming a covalent bond. And here we have two oxygen atoms. They're forming a covalent bond because they're sharing a pair of electrons. So the thing is that when it's a nonpolar covalent bond, it's equal sharing of electrons. Therefore, the charge is distributed. Remember, electrons are negatively charged, so you have equal sharing of that negative charge between the electrons. So therefore, in a nonpolar, I actually should say nonpolar covalent bond, there is no overall electrostatic charge. Now, a subtype of covalent bond is polar covalent bond. So a polar covalent bond, what happens is like sometimes some atoms, like when you have certain bonds, some pairs of atoms, one atom tends to hold on to electrons longer than other atoms. So in the case of water and the bonds between oxygen and hydrogen in water, what happens is that the oxygen tends to hold on to those electrons a little longer on average than the hydrogen atoms. Therefore, that's why oxygen, again, it's not like a full ionic charge where it's a negative one charge. But oxygen, on average, tends to have an overall negative charge. Why? Because it spends more time holding on to those electrons than the hydrogen atoms. Again, this is the key difference between polar and nonpolar bonds. With polar, you have unequal sharing, whereas in a nonpolar, you have equal sharing. So again, it's all determining, because electrons have charge, that determines whether these covalent bonds actually have a partial negative charge or not. All right, so then common misconception. So this is also a common misconception is that hydrogen bonds are the same thing as polar bond or polar covalent bonds. So the thing is that water is often used as an example of a polar covalent bond and also in hydrogen bonds, but they're not the same type of things. Polar covalent bonds are not the same thing as hydrogen bonds. Polar covalent bonds are the underlying cause of the electrostatic charges that cause hydrogen bonds. So the thing is, like a common misconception, hydrogen bonds only occur between water molecules. So hydrogen bonds are not exclusive just to water. Other molecules can form hydrogen bonds. So the thing is that hydrogen bonds is any sort of bond that it's actually a more, rather than actual transfer or actual sharing of electrons, it's due to the attraction of partial negative or actually partial positive bonds or partial positive charges on hydrogen, and it's attracted to negatively charged atoms or partially negatively charged atoms. So it's not really sharing electrons, it's a positive negative attraction. All right, so here we have water again, classic example with hydrogen bonding. But here thing is that, again, water is a polar covalent molecule. Why? Because it has all these it has unequal sharing of electrons. Therefore, the hydrogens in water tend to be partially positive. And here we have part of a glucose molecule. So as you can see, there's many carbons, there are many hydrogen and oxygen atoms. So here within glucose itself, we have a partial polar covalent bond, or actually we have a polar covalent bond. So what will happen here, again, this is also another hydrogen atom, this is another oxygen atom. And between the two, oxygen tends to hold on to the electrons a little longer than the hydrogen atoms. Therefore, in glucose, oxygen at this position is also slightly negative. So what we have here is an attraction between this water molecule here and the glucose molecule, this part of a glucose molecule. So this is a hydrogen bond. Why? Because again, there's no actual sharing of electrons between the water molecule and this glucose molecule. But since the hydrogens on water are slightly positive and the oxygen at this position in a glucose molecule is slightly negative, they are attracted, so they will attract each other. Now compared to another position in the glucose molecule, let's look at carbon and hydrogen at this position down here. So the thing about carbon and hydrogen, they tend to share the electrons equally, therefore they're nonpolar covalent. Therefore, they don't really have a strong electrostatic charge. So here's another water molecule, but there's no real charge at this point. So this water molecule has no negative charge to be really attracted to because this carbon is relatively neutral in terms of its electric charge. Therefore, it'll go on its merry way. And this is why, so again, this is just one molecule, but it's showing there's a hydrogen bond that can form between this hydrogen on the water and also this oxygen at this position in the glucose molecule. So again, I'm trying to dispel the myth and misconception that only water can do hydrogen bonding. As long as there's a partially positive hydrogen and as long as there's a negatively charged atom somewhere in the molecule, there's a potential for hydrogen bonding. So these are the main types of chemical bonds we covered in class. So what we have is is there electrostatic charge? And this is also a big difference between all of these. So nonpolar covalent, you have equal sharing of those electrons. Therefore, the charges are distributed equally between the atoms in a nonpolar covalent bond. Therefore, there is no overall electrostatic charge. Polar covalent, yes, there is electrostatic charge because that's when things start to get imbalanced and unequal. So one of the atoms, typically oxygen, nitrogen, or fluorine in terms of drugs, they tend to hold on to electrons. So here we have hydrophilic and hydrophobic. And sorry, it looks like OBS said there was connection issues. Don't worry, I am recording this, so I'll upload it later. All right, so hydrophilicity and hydrophobicity. So then what, again, hydrophilic, so again, hydro in this term means water. So does a molecule, does a chemical like water? Does it like dissolving in water? Or is it hydrophobic? Does it not like hanging out with water? Does it not like dissolving in water? Okay, what we have here, so polarity is kind of like an electrical charge in a chemical. I know it's a lot of you haven't had chemistry yet, and this is the best I can explain it to without having a big background in chemistry. Now, chemicals with a high polarity or electric charge tend to be hydrophilic. And why is that? Well, again, remember, this is why it's important to understand the polar covalent bonds in water and hydrogen bonding. So again, water does have a slight electrical charge at its hydrogen atoms and oxygen atom. So the thing is that with this partial positive negative charge, if you also have other compounds or other molecules with a positive or negative charge, they tend to be attracted to water and vice versa. Whereas if there's no charge, nothing for those partial positive and negative charges on water to be attracted to. Hydrophobic, or if a chemical does not have any sort of positive or negative charge, they tend to be hydrophobic. Why? Because again, if there's no positive, there's no negative, there's nothing for those charges to be attracted to. You need some sort of charge for this positive, negative attraction. All right, so let's talk about pH. Now, what's pH? So this is a scale to measure the proton concentration. And remember, go all the way back to, I think we did cover it last lecture, but maybe 2 lectures ago. So remember, hydrogen ions is the same thing as H+, is the same thing as a proton. So protons is when I'll use this interchangeably too. I tend to say protons or H+, but protons are referring to all these terms refer to the same thing. And why? To review, again, hydrogen ion, here's the one that has the equal amount of protons, neutrons, and electrons. What happens if you lose that electron? So what happens to the charge? Now it's +1 because that electron is negative. Lose that in a negative charge. Now it's 1 minus 0. That's positive. And remember that in our universe, the vast majority of hydrogen atoms have no neutrons. So when you lose that electron from a hydrogen atom, you're just left with a proton. So this is why you often hear proton just referring to a hydrogen atom that's lost its electron. Now, why did I bring this up? So let's talk about acids. So here I'm going to show you 3 types of acids, and what do they all have in common? Notice that they all have hydrogen ion atoms on here. But the thing is that right now, even though they have the sign, I show a positive sign and negative sign here, right now they're bound to their original molecule. Now, what happens if you throw these acids in water? Well, when you start throwing them in water, these protons start popping off. These hydrogen ions start dissolving and dissociating from the original structure. So now you're releasing all these protons, all these hydrogen ions into the surrounding solution. So this is what makes something an acid. So again, what happens is that when you put it into a solution, what happens is that now it's releasing all these protons into that solution. And this is critical to understanding pH. Now, pH scale indicates hydrogen ion concentration. So basically, how much hydrogen ions are located in this solution, how dense it is, how packed full of protons and hydrogen ions is a solution. So as you can see here, there ranges like, and I can actually go further past this. There is a such thing as negative pH, but in terms of the body, notice that all the bodily fluids like urine, saliva, milk, blood over here, it tends to be around here, although things like stomach acid, very acidic. So we are talking about hydrogen ions. So again, things that just because it's neutral, common misconception and pitfall, a neutral solution as water, such as water, has no hydrogen ions, has no acidity whatsoever. That is false. So again, the thing is that a neutral solution can have hydrogen ions, but tends to have equal amount of base or hydroxide ions that balance out that pH. So it just refers to concentration. Now, something that's very acidic, compared to something like water, it's going to have so when they say orders of magnitude, they're usually referring to something times 10. So every step in the pH scale is actually 10 times. So you get 10, hundreds, thousands. Yeah, so I'm actually not showing it to scale here. What I'm getting at is that the more acidic something is, the more protons, the more hydrogen ions you have in that solution. And when you have something basic, so again, common misconception is that if something's neutral, it has no hydrogen atoms. Something that's very extremely basic, it still might have some hydrogen ions, but far less than a neutral solution, and way, 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 way less than something that's very acidic. So that's what we were talking about with the pH scale. It just refers to hydrogen ion concentration. So here we have the acidic range. So again, acidic meaning that it has, in terms of compared to neutral with pure water. Again, pure water does have trace amounts of hydrogen ions, but something that's more acidic than water has a higher concentration of hydrogen ions and protons than water does. So as you can see, stomach acid is much more acidic than pure water and urine and saliva. Therefore, stomach acid has a higher concentration of protons than urine, stomach, and water. Now what we have here on the opposite side of the scale, we still have pure water here at pH 7. So that's a useful value. pH 7, this is considered neutral, so that's where pure water should be. Now over here we have the basic side, and what we have here is that this is, again, neutral does not mean a complete lack of protons, but something that's basic has less concentration of protons than a substance that's neutral like water. So things like 1 molar sodium hydroxide, and again, if you don't know what molar is, if you're taking chemistry 161, you're going to find out really, really soon. So a molar just refers to it's just a unit of concentration, so what's happening here? This is so what I'm getting here is that a basic solution has less or fewer hydrogen ions than something that's neutral. But you might want to also pay attention down to this part right here. So notice that this is, whenever you see these double brackets, it refers to concentration. So what we're seeing is the concentration of hydrogen ions, aka protons, right? So as you can see, this is going into the negative exponent. So this is actually a concentration that's going, so from here, this is a concentration of 1 molar, and this is actually 0.1, 0.01, 0.001. And this is why, as you're going from here, it's not equal to 0. It's very tiny. But as the pH goes up, you notice that it's getting the order of 10, 1/10, 1/10, 1/10, 1/10 less hydrogen ions than the previous notch on the pH scale. Okay, so notice that blood, compared to other types of fluids, and I did ask this in the packback. Oh, and actually, speaking of packback, there was a really good question asked during office hours. So there are over 300 of you. Some of your questions may overlap, and that's fine. Like, it's going to be really hard to come up with 300 completely original questions. And do you have to read all 300 questions? No. So you are able to kind of filter based on like what's new, what's unanswered, what's hot. So you can answer what you like. All I'm looking for by the end of this upcoming Sunday is just one question that's above that 30. And as you can see, it's pretty easy to hit that 30 curiosity points threshold. But again, if you somehow miss it by a few, you can edit and add a little more to your question to push it over that 30 curiosity points threshold. So again, don't feel like you have to read all those questions. You can answer the ones that you find the most interesting. All right, so back to blood. So notice that blood, it has a very, very narrow range. It's actually 7.4 plus or minus 0.05, not 0.5, 0.05. So it's the normal range of arterial blood, and that, in other words, blood that's in your arteries, is between 7.35 to 7.45. It's a very, very narrow range. Now compare that to something like urine. So urine, the normal, in terms of medicine, the normal range can actually range like from roughly around 4.5 to 8. But say you're getting up in the morning, and if you just take random urine samples from everyone, urine is actually more around this range. But if you're depending on your diet too, that can also cause your urine to go toward more the basic side. Oh, and basic and alkaline are used interchangeably. So acidic is acidic, but when you heard that term alkaline, it refers to this range of the pH scale. So urine tends to be acidic, but the normal range of urine tends to be around this one, this pH scale, around roughly 4.5 to 8. But why is this a normal range? Well, the thing is that this is household ammonia, which you won't find normally in your body. But there are certain bacteria, if you have they call a UTI, a urinary tract infection, some bacteria actually like to take this compound in your body that you produce all the time. The compound is called urea, and they do something called urea splitting. And what happens when urea splitting occurs in the body, or actually when bacteria do this, it splits urea into another metabolite and ammonia. So if a bacteria is converting urea to ammonia, this might cause the pH of urine to get more and more basic and alkaline. So if we see like, oh, your urine pH is 9, it might indicate that you might have a possible infection with one of the bacteria that converts urea to ammonia. So this is why it's kind of important to know pH. Now, how is pH determined? So this involves algebra 2. It involves logarithms. So this is one of the toughest things when you're first learning pH. The more acidic something is, the smaller the pH number. And why is that? Well, as you saw that the concentrations, they're always 10 to either 0 or 10 to the negative something. So they're actually not just like, they're actually a very, like 10 to the 0 is 1. But 10 to the negative 1, that's 1 over 10. 10 to the negative 2, that's 1 over 100. So the thing is that if you take a logarithm of a fraction like that, it's going to be negative. So the more negative, the smaller the fraction, that's going to cause a more negative number. But the thing is that with pH, this negative sign, this actually flips it the other way around. So this is why when something's very basic, has a very low proton concentration, that's why it has a high pH. Whereas something that's acidic that actually technically has more protons, this is why the pH tends to go down. So it's funny, like the more acidic something is, the lower the pH. And this negative sign is the whole reason behind that, kind of like counterintuitive part of pH. So again, the lower the pH, the more protons you have in the solution. The more protons you have in the solution, the more acidic the solution. But this is the part that's kind of like counterintuitive. This negative sign right here, and lower pH means more acidic, more protons. Okay, so let's talk about biological macromolecules. This will be the kind of like I'll wrap up chapter 2 with. So there are 4 main categories. There are the carbohydrates, lipids, proteins, and nucleic acids. So thing is that what's a good mnemonic to know memorizing all these 4 broad categories of biological macromolecules? Well, just pick up anything. Well, I don't have food in my room right here, but just look at the back of a nutrition label. And why did I bring this up? Well, carbohydrates, those are definitely on a nutrition label. Same with lipids, aka fats. These are also on a nutrition label. And then you have protein. Now, nucleic acids, why do I have this asterisk? Well, the thing is that nucleic acids aren't on a nutrition label, and I will get to why they're not on there at the end of this lecture. But 3 out of 4, just remember when they talk about, or like if you have someone who's really into fitness, they might talk about their macros all the time. So they're talking about macronutrients and biological macromolecules. Yeah, so then 3 of 4 of them are those macronutrients they're always talking about. Now, first, let's start off with carbohydrates. So carbohydrates, so there are many types of carbohydrates, but these are broad categories of carbohydrates. So monosaccharides, and remember, I love those prefixes, I love those root words, and why? Because they tell you the bigger vocabulary you have of them, the more you can figure out new terms. So mono, just like a monorail, right? Like ours that's never getting built. That's the monorail. So again, it just means one. So monosaccharides just means one sugar, one carbohydrate. So things like glucose, fructose, and galactose, these are monosaccharides. Whereas disaccharides, so di meaning two, like something like when a path diverges, it splits into two. So disaccharides, that means two. So then things like sucrose, maltose, and lactose. So each of these is made out of two monosaccharides that are linked together with covalent bonds. All right, so polysaccharides. So polysaccharides means many. So again, poly actually this is used all the time in medicine. So when you have something like polydipsia, that means many thirsts. That means you're very thirsty. Or polyuria, that means you're producing a lot of urine. So polysaccharides means many saccharides linked together. So you just take a bunch of monosaccharides, link them together in a long chain, and you have a polysaccharides. So things like glycogen, starch, and cellulose, these are all polysaccharides. So they're long structures made up of a bunch of monosaccharides linked together with covalent bonds. Now, structures and properties. So here I'm showing glucose. And actually, again, I'm pretty sure most of you have not had organic chemistry yet. So the thing is that this is often what you might see in your textbook, especially OpenStax. And this is when I say like OpenStax is kind of like alternates between very easy and very hard. So some of the pictures, when you see these and you're like, okay, what are these angles and sticks and lines? So when there's an angle without really explicitly saying what the atom is, it tends to be carbon. So that's like something you'll see, especially if you're taking organic chemistry, you'll see these structures all the time. So what we have here is glucose. It has a bunch of carbons, and I'm not showing all the hydrogens either. So this is just one way of showing a molecule in a simpler manner. But what I'm getting at is that polar covalent bonds. So remember that oxygen tends to hold on to electrons a little longer than hydrogen. So you have all these partial negative charges on these oxygen atoms on glucose. And therefore, all these water molecules are attracted to it, right? So it is very hydrophilic because of these partial negative charges and partial positive charges on glucose. This is why glucose and simple carbohydrates dissolve readily in water. Now, the thing is that carbohydrates can be also linked. So glucose is a monosaccharide. Here we have glucose, a monosaccharide, and we have fructose, another monosaccharide. So now we're going to form a link between them. So they're going to form a polar covalent bond. But this is the thing. They have all these atoms right here, but the excess atoms pop off. So what happens is that glucose and fructose are linked via oxygen atoms, but the extra oxygen and two hydrogens pop off. What is H2O? So H2O is water, right? So this is why sometimes it's called a dehydration synthesis reaction. So you're producing water, but you have it's also synthesizing a new bond between these two former monosaccharides. So now you have sucrose, and this is why sucrose is a disaccharide. You had two monosaccharides, you joined them together, now you have a disaccharide. All right, so disaccharides, there are many disaccharides, but the ones I want you to know, especially if you're continuing on to PHYL 142, so glucose plus glucose equals a disaccharide called maltose. Now glucose plus fructose, we just covered this, and you might be familiar with this, sucrose, aka table sugar. So sucrose is not a monosaccharide, it's actually glucose plus fructose. Now then you have glucose plus galactose. That might be familiar to you, carbohydrate called lactose. So this is another pitfall that's often, so galactose and lactose sound very similar, right? So some people think that galactose is a disaccharide because it's a longer word. But what we actually see here, lactose is actually the disaccharide, and the longer word is actually the monosaccharide. So galactose, that's the monosaccharide. Galactose plus glucose equals lactose. So these are the three I want you to know. All right, so carbohydrate. So here we have one monosaccharide, and then when you have two monosaccharides linked together, we have a disaccharide. And what do we have here? Oh, 3, 4, 5, 6, 7, and more. So what we have here are many saccharides linked together. This is why when we have many carbohydrates and monosaccharides linked together, we call it a polysaccharide. So again, this is what we call a polymer. So a polymer is something that is a chemical structure that can repeat over and over and over again, theoretically until infinity. So you can keep on building upon this original polysaccharide. So things that macromolecules, they have these basic things like monosaccharides, but the things that macromolecules can form build upon each other using these basic units and form more and more complex structures. It's kind of like Minecraft. So the thing is that polymer is a long chain of molecules with a repeating structure, and this repeating part is a critical component in what makes something a polymer. So say this is before social distancing, okay? So this is one person, right? So what happens if you have a theoretical polymer of people? Well, again, a polymer is a chain of something, molecules, with a repeating structure. So here is what a polymer of people might be. So notice that they're all linked together and they're repeating over and over. And theoretically, if you add more and more people, you can add a lot more and more to this polymer of people and this chain until infinity. So this is what I'm referring to when I talk about a polymer. So things that carbohydrates, proteins, and nucleic acids, these form polymers. But things is that fatty acids and lipids do not form polymers in your body. So this is what I'm getting at. All right, back to carbohydrates. So here we have a polymer, we have a polysaccharide and polysaccharides. So we have many of these monosaccharides linked together in a covalent linkage. So this is actually what we call amylose. So when you have a straight chain of carbohydrates linked in a polysaccharide, this is amylose. And actually one type of amylose, but just know at this level, amylose is a straight chain with no branches. But when you have starch, now starch, you digest starch, but you don't produce starch unless you're a plant. But starch, compared to amylose, has a little more branching. Now, glycogen, this is not what you can produce. So the glycogen is produced in your body. This is actually a storage form of glucose in your body, and especially in your liver and skeletal muscle, this is where you'll find a lot of glycogen. So what's the big difference between glycogen and starch? Well, very roughly, glycogen tends to be even more branched and complex than starch that you find in plants. But again, glycogen, why do you want to build up all this glycogen? Say you're eating a meal, but you want to save all those carbs for energy later. And this is one of the concepts behind carbo loading for athletes. So since you're storing glycogen, you're kind of building up these energy reserves, but once you're actually doing physical activity or you need that energy, you can break down that glycogen and release all these simple carbohydrates to release into your bloodstream and fuel your cells for exercise or whatever physical activity you're doing. Okay, so finally we get to top hat. So these reactions I'm showing here, look at this middle reaction. Glucose plus fructose equals sucrose. In terms of that reaction, glucose plus fructose equals sucrose. What best describes this reaction? So let's open it again. Okay, let's see how everyone did. Okay, so we can see that there are a variety of responses, and so is it anabolic, catabolic, or synthesis reaction? So the thing is that this is correct. It is anabolic because remember, anabolic is building something new, right? So you're building new bonds. So what we have is a new bond between glucose and fructose. It is an anabolic reaction. It's not a catabolic or decomposition because what we have two reactants and it's building a new molecule, it's building new bonds. Catabolic and decomposition means it's breaking apart. It's not an exchange because we didn't have different partners swapping different groups. And it is a synthesis reaction because we are forming new bonds between. This is why it's also called a dehydration synthesis reaction, when you go from glucose plus fructose into and synthesize a new sucrose molecule. All right. Okay, so sorry about that. So let's go back to our lecture. So now we're done with carbohydrates for now. Again, if you're going on to PHYL 142, we're going to cover carbohydrates again. But this is all you need to know for now. So then let's go down next to lipids. So lipids, these are also known. So this might be a new term for you. Like I think in different countries, like especially in like Canada or especially European country, they like to use the term lipids, but they're also known as fats. So fats, oils, and waxes, these are all types of lipids. So not the red part of this meat, but look at this meat. It has all this white stuff, right? That's all fat. Or if you're a vegetarian or vegan, this is also a very tasty fruit, right? But it also has a lot of fats as well. And even oils, these are lipids as well. And butter, that's also a lipid. So these tend to be hydrophobic. So if you try to like create a dish with a lot of butter or oil or very greasy, can you just like wash it off with just plain water, no soap, nothing else? No, you're going to have a hard time, right? So the thing is that lipids have a hard time dissolving in water. And this is why do they tend to have a hard time dissolving in water? They tend to have either long or very complex hydrocarbon backbones, and it sounds like what it means. So that means their base structure, their underlying chemical structure, tends to be mostly carbon-carbon and hydrocarbon non-polar covalent bonds. Again, non-polar covalent bonds, meaning that the non-polar that there is equal sharing of electrons. Therefore, there is no strong electrostatic charge on these atoms that are forming this non-polar covalent bonds. In other words, these hydrocarbons do not tend to have strong electrical charges. Now, why does that mean? So classic simple, when we talk about like things that are lipid derivatives, they tend to be most of the time we're talking about a compound or a molecule that's derived or made from a fatty acid. So this is the structure of a fatty acid. So what do we have here? So what we have here with the fatty acids, they're long chains of carbon and hydrogen ending with a carboxyl group. So in this molecule right here, these black balls right here, these are carbon atoms. In these light blue colored balls, these are hydrogen atoms. These are two oxygen atoms right here. So what you have is a bunch of carbon and hydrogen in a long chain ending with carbon and oxygen. So here we have this is what we call a carboxyl group. And this is another thing with my slides. When I have something that's italicized, that's something I think that might be a little too advanced, because carboxyl group is more like, especially if you're going to organic chemistry and taking that, you'll definitely need to know what a carboxyl group is. But carboxyl group, when you have a group in chemistry, it's usually like atoms tend to form these certain structures repeatedly in nature. So this is something you often see in nature. When you have these two oxygen and a hydrogen atom linked to a carbon, this is what we call a carboxyl group. And these are what we call carboxylic acids. So this is what makes it an acid. So these are mostly nonpolar except for this carboxyl group. So here we have a water molecule. So the thing about water molecule, again, remember, due to those polar covalent bonds and unequal sharing of electrons, the hydrogen tends to be a little positive. So that these atoms, oxygen atoms on a fatty acid, these tend to be partially negative because they also have their own polar covalent bonds in a fatty acid. So water does like to associate with this part of a fatty acid. Now, what happens when water gets to this part? Well, the thing is that these hydrocarbon chains right here, these carbon-carbon, hydrogen-carbon bonds, they are nonpolar, so they don't really have an electrical charge. With no real electrical charge, there's no real attraction between water and this part of the fatty acid. So as you can see, most of the structure of fatty acid is pretty like water could care less. There's no real electrical charge, so it can't really bond to these fatty acids. So these are why fats in general have a hard time dissolving in water. There's nothing really for water to be attracted to. Okay, so then fatty acids. So fatty acids may be saturated and unsaturated. We get to that in PHYL 142. Again, if you don't know what single and double bonds are, it's kind of hard to explain at this point. But thing is that these bonds, or actually don't worry too much about these, but these come into, especially if you're going to nutrition, you have to know about saturated and unsaturated fats. So glycerides, so these are fatty acids, but they're attached to a new molecule called glycerol. So what is glycerol? So the common misconception, glycerol is not a lipid in itself, but fatty acids and lipids tend to attach to glycerol, especially when you're forming a triglyceride. So common misconception, triglyceride does not mean 3 glycerols. When you have that monoglyceride or diglyceride or triglyceride, it's referring to the amount of fatty acids that are linked to glycerol, not the glycerol molecule itself. So triglycerides, also known as triacylglycerols. So here's glycerol, and again, glycerol is not a fatty acid, but what happens in a triglyceride, you can actually get 3 different fatty acids and then covalently link them with chemical bonds to a glycerol molecule. So this is when you get a triglyceride. So again, it's not 3 glycerol molecules, it's 3 fatty acids plus 1 glycerol molecule. This forms this entire structure right here is what you call a triglyceride. Okay, so steroids. So when you think of steroids, you probably think of this, right? So steroids, you probably think, so again, but what is a steroid in terms of macromolecules? So steroids refer to any sort of chemical compound that has this basic backbone. Thing is that sometimes you may have different atoms and chemical groups added to different parts of the steroid, but the thing is that steroids all come from an original structure, and this might sound familiar, cholesterol. Cholesterol always has these 4 rings. So in Martini, it gives example of different steroids, but this is a class of lipid compounds. They are not derived from fatty acids directly. Yeah, it's actually a more complex pathway called the mevalonate pathway, but that's very biochemistry. Don't worry about that. But steroids, just know that they are lipids and they look way different. Again, instead of being a long chain of carbons and hydrogens, they have this complex ring structure. So these tend to be generally hydrophobic, and these are very important for lipid metabolism, and especially when we get to PHYL 142, there's a whole class of hormones called steroid hormones, and all of them generally look like this structure right here. So this is an example. So steroids are all derived from cholesterol, and here we have estrogen, actually more accurately estradiol, which is the primary estrogen in the human body, and testosterone. But look what they have in common. They all have that 4-ring structure. So again, I mean, this is the way I learned it when I first was starting out. It's 3 hexagons linked to a pentagon. It does have more carbon and hydrocarbon chains right here, but this A, B, C, D ring right here, these 4 rings, the 3 hexagons and the pentagon, pretty much found in the majority of steroids with a few exceptions. All right, so then let's move on to another category. These are phospholipids, and this is phosphate. So here we have a phosphorus atom linked to, covalently linked to 4 oxygen atoms. So what we have here is that because the oxygen atoms, and it's kind of hard to see here, it's very tiny, but these have, so a phosphate group has a -3 charge. It does have an ionic charge. So with this negative charge, all of these water molecules are going to love it. Remember, water does have its own charge, so the positive hydrogen atoms of water are going to be attracted to a phosphate group. Now, here we have more thing. So here we have an amine group linked with the hydrocarbon tail. So things that this part with this phosphate and this polar bonds right here, water loves it. There are electrical charges for it to grab onto, so this compound is able to dissolve freely in water. Now, this is what where the phospho part of a phospholipid comes from. Now, the thing is, this part is hydrophilic, but when you have a phospholipid, now you have these long hydrocarbon tails. So remember, these look like those fatty acids, right? And actually, they come from those fatty acids. So remember that water does not like this part of a fatty acid. Why? Because carbon-carbon and hydrocarbon bonds don't tend not to have a strong electrical charge. There's nothing for water to be really attracted to. So therefore, this part tends to be hydrophobic or lipophilic. It likes other fats as well because they don't really have a strong electrical charge to be attracted to or repel. So again, hydrophilic and hydrophobic, and then you have something called amphipathic. So this is the thing about those phospholipids. Notice that it's the same overall molecule right here, but it has one side of it that likes that's hydrophilic and likes associating with the water, but it also has another big part of it that's hydrophobic. So hydrophilic, meaning you like water, it dissolves easily in water. Hydrophobic, meaning not easily dissolved in water. So when you have amphipathic, so it's like an amphibian. An amphibian lives on both land and water. So amphipathic molecules, they associate with both hydrophilic and hydrophobic compounds. So this is like, it's kind of like that friend that's, you might have a friend that kind of likes to associate with two different social groups that don't really get along. So they're kind of like that floater. They like to hang out with one side, but they also like to hang out with the other side as well. So amphipathic molecules are kind of like those people who like to hang out with two separate crowds. Okay, so let's talk about proteins. So proteins, these are very, very diverse. Like I know it's kind of like fudging it, but there's so many different proteins. They do so many different purposes that you can't memorize a whole list of proteins at this level, or in your lifetime. So proteins, they contain carbon, things like lipids and carbohydrates, they have carbon, hydrogen, and oxygen. But notice that proteins, they have nitrogen. And this is critical, especially when you're continuing to fill 142 and metabolism. So amino acids, these are basic building blocks of proteins, and 20 standard amino acids. So amino acids, just like monosaccharides are the building blocks of polysaccharides and complex carbohydrates, amino acids are the building blocks of proteins. So 20 standard amino acids, so things that they will probably learn, I think it was a Bio 265 or 275. I think they have you still draw or memorize the structure of amino acids. If you have a very good teacher, or if you go to grad school, you might learn about selenocysteine, but don't worry about that. Just know the standard amino acids for this class. So protein structure, this is showing part of an amino acid. So there are long chains of amino acids. So this, again, the carboxyl group, just like in the fatty acids. So this is why amino acids are called acids, because they have this carboxyl group. Now, this part right here, this is an amine group. So nitrogen linked to 3 other groups. So this is why it's called amino acid. It has the acid part that can pop off that hydrogen to release a proton. It also has an amine group. Now, they have a common basic structure. So all 20 types of the basic amino acids, they have this underlying structure. But this side chain, so this is why it's an R. This is the difference between the 20 different amino acids. So there are many different types of atoms and bonds that can be formed here. So this is the part, there's 20 different things that can fit into this part. But all amino acids have the carboxyl group, the amine group, and this carbon that anchors all of them together. So then so this is one that shows 21 amino acids. Selenocysteine is the one that tends to be the one that's omitted from other textbooks. But yeah, so just know all of them, selenocysteine is kind of like the special one. Not that it's not important, it's actually very important, but don't worry about that too much. All right, so peptide bonds. So things that, here we have 2 amino acids. And what happens is that between the carboxyl group and the amine group, you have this linkage that forms. So this is what we call dehydration synthesis. Just like we had with carbohydrates, you have 2 amino acids. They're now linked together. So when you have this new bond between a carbon and a nitrogen atom, this is what we call a peptide bond. So a peptide is not this amino acid. It refers to the special bond between carbon and nitrogen between amino acids. Now, again, peptide bond is this new bond. So again, between amino and carboxyl group, again, a lot of you haven't had organic chemistry. Don't worry too much about that. That's the more chemistry definition. For our definition, just know that this is the bond between that carbon and nitrogen that links two amino acids together. So when you have many peptide bonds together, this is called a polypeptide. And I know I'm going over time here, so if you have somewhere else to go, you can leave right now, but I'm going to try to finish up this so that we can continue on to chapter 3 on Friday. All right, so again, 20 asterisk of different amino acids. That's not including selenocysteine. So when you have one amino acid and then you have another amino acid, remember they're linked via peptide bonds. So peptide bonds, so what happens? We have many peptide bonds linked together. So this is why when you have a long chain of amino acids together, this is why you call it a polypeptide. So again, peptide is not the same as amino acids. They're related, but the peptide part is referring to the bond. Amino acid is referring to that individual building block that's being linked by peptide bonds. So again, polypeptide, chain of amino acid linked with peptide bonds. Again, they're related, but not the same in terms of amino acids and peptide bonds. All right, so here's a protein primary structure. So what we have here, all these amino acids, and they are amino acids, but they're linked together in this long chain. So I like to do an analogy. So this is actually insulin, but when you see a picture of insulin, they typically don't show all the atoms of the insulin linked together in this. As you can see, it's very complex. But here I show a picture of hair. So like when you ask someone to like, oh, show me a picture of hair, they're probably not going to show you the up close, in depth. So this is what you call electron microscope scan. So what happens is a very detailed, high resolution, ultra magnified resolution picture of hair. The thing is that with insulin, you're probably not going to see this picture. So then you have protein secondary structures. So to take that hair analogy, it's like when you take a strand of hair and then start building it and start to style it into different ways. So you can take a polypeptide and you can style it and form these other structures as well. So if you take a polypeptide and kind of coil it and it starts to make another coil, this is what we call alpha helix and beta sheet. Don't worry too much about this. Again, if you're taking molecular biology, you're going to cover this definitely. And sorry, it's lagging right now. All sorts of technical difficulties today. Okay, so maybe that's a sign for me to call it a day right now. So sorry about all the technical difficulties. I'll try to figure out what's going on with Top Hat, and I think just like PowerPoint's just throwing up right now. So I will continue on Friday. We'll pick up where we left, and I'll try to get into cells by then. So we are a little behind, but we'll try to catch up really soon, all right? So see everyone on Friday. Office hour starts at 3, and I'll see you there.